Okay, moving on. Here we go. So it is my pleasure to introduce to you today Dr. Ryan Logan, who at the time, I don't want to minimize this, sorry, I'm setting up my technology here. I want to be able to see him. Who at the time was an assistant professor of psychiatry at University of Pittsburgh. And since then, he's joined my department in the Department of Pharmacology at Boston University School of Medicine, where he was hired at the associate professor level for good reasons, which I'll get into in a minute. So Ryan is associate professor and director of the Laboratory of Sleep Rhythms and Addiction. He received his undergraduate degree in psychology at the University of Maine, PhD in neuroscience at the University of Maine, where he worked with Dr. Alan Rosenwasser, who's a long-standing member of IBANGS, and subsequently trained in 3 separate postdocs, which I really think added to the diversity of his research program, including Deepak Sarkar at Rutgers, where he studied immunology and neuroscience, Dr. Alyssa Chesler at the Jackson Laboratory, where he gained experience in mouse behavioral genetics, computational biology, and genomics. He then went on to do a postdoc with Colleen McClung, who has also attended previous IBANGS meetings in molecular neuroscience, studying circadian rhythms and the intersection with drug addiction. So he was assistant professor from 2015 to 2020 in psychiatry there as well. And then in January 2021, he started here officially at Boston University School of Medicine. Dr. Logan has earned several awards over many years, career development awards, including NARSAD, a K Award to study IMPASS-2 in cocaine addiction. A few years ago, he got an R33 to generate novel mouse tools for understanding circuitry of circadian molecular mechanisms of addiction. More recently, he received an R01 to study sex differences in molecular rhythms from human brain tissue, and just recently had a first author paper accepted in biological psychiatry that addresses this question. Many other awards, including a CBRA R21. I think he's going to talk a lot about this today, to look at whole brain imaging of mouse brains, and to identify cell types, projections, and circuits throughout the mouse brain. So this uses machine learning and artificial intelligence approaches. Another award he was recently awarded was cell type specific role of circadian dependent transcription in fentanyl-induced synaptic and behavioral plasticity. So he's really has a diverse program studying multiple drugs of abuse and intersection with addiction. He's won multiple awards in addition to this prestigious IBANGS award at various societies, including RSA, SOBP, SFN, SRBR, and more recently, ACMP Travel Award. I don't have time to list all the publications he has, more than 50 very impactful journals. The overview of his research program is, the unique aspect is the intersection between basic research and the clinical component, where he studies both rodents in vivo and tissue, as well as human tissue from patients to understand the intersections between sleep and circadian rhythm and substance use disorders, as well as mood disorders. I've mentioned some of the cutting edge techniques that he uses. He'll talk more about those. And I will note that he's a highly collaborative investigator. And I think this speaks to the fact that he has very high level of scientific enthusiasm, but also he's genuinely a really nice guy and colleague. And I'm sure that my department will come to realize this over the coming years. And so, so without further ado, please join me in congratulating Dr. Ryan Logan for his accomplishments and winning this Young Scientist Award for the IBANGS meeting, and welcome him in delivering his talk today, which is entitled Novel Sleep and Circadian Mechanisms, Linking Rodent Humans and Opioid Addiction. And at this point, I think we'd be handing you a plaque, but we don't have that today, so there you go.
Thank you, Cameron, for that wonderful introduction. And I just want to thank the IBANGS committee for selecting me for this award. I'm truly honored. I've been attending IBANGS, I believe, since 2012. with a few misses in there, but it's a fantastic organization and conference. So I went back and forth a little bit on, can everybody see this? Okay. So I went back and forth a little bit about what I was going to actually talk about today. And I decided that I'm going to talk about all unpublished data. And hopefully some of this is brand new, and hopefully it's well received, but definitely open to feedback. So this is just conflict of interest disclosure. None of these impact the data that I'm going to talk about today. So the title of my talk, as Cameron mentioned, is Novel Sleep and Circadian Mechanisms Linking Rodents and Humans in Opioid Addiction. And I just want to start out that a lot of this work started when I was in the Department of Psychiatry at University of Pittsburgh, and I joined the Department of Pharmacology at Boston University as recent as January 1st of this year. I do not recommend moving to young kids and your lab in the middle of a pandemic. So the approach that we take in the lab is really this kind of translational neuroscience approach. What we're really trying to do is model human disease or aspects of human disease in our rodent models. And we take a number of different approaches to actually do this. And we've published across each one of these arenas. And mostly what I'm going to be talking about today is our work, most recent work using human postmortem brain, and how we've tried to integrate that across different levels of biology in our rodent models. Another active arm of research in our laboratory is both developing and employing new tools to look at cross-species integration at the molecular and cellular level. And so we have a lot of work in computational biology and bioinformatics. The framework that this fits in is really focused on the role of circadian rhythms in sleep, both in how circadian rhythms and sleep modulate and contribute to cognition, mood, and reward, but how circadian rhythms in sleep might be a mediator or direct modulator of both the emergence, development, and progression of various psychiatric disorders. And in our animal models, we develop and design experiments looking at both kind of the environmental pressures or factors that can impact the neural circuitry and behavioral phenotypes that we're interested in, and also the genetic impact. So both kind of at the pure genetic level, where we're trying to discover new genetics, new variants that ultimately regulate rhythms and sleep at the intersection of reward and mood, but also how circadian genes, which I'll talk about in a second, are really integral in these processes that we know are important for psychiatric disorders. And of course, we're also interested in developing new treatments, intervention approaches using our preclinical models stemming from our human work. So what is the circadian rhythm system? Circadian rhythms are near 24-hour cycles in every biological, physiological, and behavioral process. They're endogenously generated, but they can be entrained or disturbed by external and internal factors. So external factors such as food and light, and can be entrained by internal factors such as hormones. It's typically construed as a hierarchically organized system. So the orchestrator or the conductor is located in the suprachiasmatic nucleus of the anterior hypothalamus. And the suprachiasmatic nucleus sends a number of projections to other brain regions, but also peripheral organs to both synchronize and orchestrate circadian rhythms across the whole body. Another important point is that almost every cell in the body expresses the necessary machinery of molecular clocks. And so I'll take you through this fairly briefly. But light, oh, I lost my mouse. Light comes in through the eye and then is transmitted through the retinohypothalamic tract into the suprachiasmatic nucleus. And then the suprachiasmatic nucleus, here, let me get my mouse back, sorry. Light comes in through the eye, travels to the suprachiasmatic nucleus, and then the suprachiasmatic nucleus relays this light information to the rest of the brain and the rest of the body, mainly through hormone regulation and sympathetic and parasympathetic nervous system, but also body temperature. Other things can impact clocks throughout every organ in our body, including food and exercise. Nearly every cell in the body expresses what are called circadian genes. And these circadian genes form a series of positive and negative feedback loops that control time-dependent gene transcription. And so the positive loop of the molecular clock is driven by the heterodimerization of two transcription factors, one called clock and the other called BMAL1. A homologous transcription factor called MPAS2 can also bind BMAL1, and I'm going to be highlighting some work that we've done in MPAS2 today. But these transcription factors heterodimerize and are recruited to various enhancer elements called E-boxes in the promoter of a number of genes. And some of those genes are these purr and cry genes that once translated in the nucleus are shuttled back in the cytoplasm, are shuttled back into the nucleus to interact with these BMAL1 heterodimers and shut down clock or MPAS2 dependent circadian transcription. And there are another number of other loops that the field has discovered over about 20 years of research, showing that different signaling mechanisms can impact both the timing, amplitude, and phase of these molecular clocks. So we know from a growing clinical literature that there's these bidirectional relationships between sleep rhythms and addiction. We know that chronic disruptions to sleep and rhythms can contribute to substance use, independence, vulnerability. So that's one end of the spectrum. And then the other end of the spectrum is kind of this reciprocal relationship where chronic substance use can detrimentally impact sleep and circadian rhythms. And we believe, and other labs believe that are studying this topic, that these persistent, long-lasting changes to sleep and circadian rhythms actually increase the vulnerability to relapse. We also know that a majority of the patients with opioid use disorder report difficulties in sleep and the timing of sleep. And so this was a statement that was put out by NIDA several years ago, where sleep deficiency, irregular circadian rhythms, and poor sleep quality is highly prevalent in patients diagnosed with opioid use disorder. And more than 75% of patients report serious sleep and circadian problems prior to and also during treatment, which I think is a really important point. It also is believed to contribute to craving and relapse, as patients often use opioids to alleviate or mitigate these states of hyperarousal or agitation and these sleep disruptions. And so we think these are intertwined. So I'm going to focus on two major nodes that kind of underlie this biological relationship between sleep rhythm disruption and addiction. One of which is the nucleus accumbens, a major neural substrate of reward. It assigns salience or motivational value to stimuli, and it's critical for regulating motivated behavior. The nucleus accumbens forms a circuit with the prefrontal cortex. We know the PFC is a major neural substrate of decision-making, guides motivated and goal-directed behavior, and it's also critical for regulating impulsivity. And we know that sleep and circadian disruptions ultimately in humans lead to decreased activity in the prefrontal cortex and increased activity in the nucleus accumbens. So we wanted to start to understand the cellular and molecular mechanisms underlying these relationships between circadian rhythms, molecular rhythms, and addiction biology in the brain. So what we decided to do, since there's extensive amount of work coming from various labs, including Colleen McClung's lab, looking at the impact of molecular clocks on reward and motivated behavior in rodent models, we wanted to actually try to bring this to humans. And so what we did was we formed a cohort of 20 subjects, unaffected control subjects that were matched on various components of things that we should care about when we're doing human postmortem studies, such as postmortem interval and RNA quality and age and sex and so forth. And we matched them to 20 subjects with opioid use disorder. And then we isolated various brain regions from this postmortem tissue, including the caudate, the putamen, the nucleus accumbens, and dorsal lateral prefrontal cortex. And I'm only going to be talking about the nucleus accumbens data today. So our first question was fairly simple. Are transcriptional rhythms altered in the brains of people with opioid use disorder? So should we care about molecular clocks in the human brain in the context of opioid use disorder and chronic opioid use? So how do we do that? Well, it's a bit of a challenge, right? In rodents, we can sacrifice and remove tissue at any time of day across multiple hours of the day and then recreate the biology by fitting various curves. In humans, obviously, that's a little more difficult. So we use this approach that we've published on several times called time of death transcriptional rhythm analysis. And basically what you do is you take the time of death of the subject, and then you normalize the time of death to Zeitgeber time. So we basically anchor the time of death to sunrise and sunset on the day they died. And we also account for the latitude and longitude of where they actually died. Now, we can't control for a lot of different things, such as the amount of light that person was exposed to prior to death, but we only select subjects with a postmortem interval of 2 to 4 hours. Okay, so these subjects are not people who were found dead days later or had postmortem intervals of 24 hours or longer. And then you arrange those subjects across a clock. And then you can take each one of those subjects and start to build curves based on the time of death. And you can fit curves relative to day and night on that date of death. Okay, so we're recreating a molecular rhythm using time of death in a cross-subject design. So we actually borrowed this and improved on this analysis from Huda Akil's and Biff Bunny's group, where they published in PNAS several years ago, looking at circadian patterns of gene expression in human brain and how they're disrupted in major depressive disorder. And since we've published on the effects of aging in a large cohort of about 270 brains in the human prefrontal cortex. We've also published in the context of schizophrenia. And most recently, we've published looking at male and female differences and how those contribute to molecular rhythms in the human cortex. So what we found in our cohort of unaffected comparison subjects, so if you take the top 250 transcripts that are quote unquote rhythmic, and you order them by Zeitgeber time, so basically by phase, what you'd expect to see is what we saw, these nice blue and yellow waves that signifies genes are changing as a function of time of death or Zeitgeber time. If you take those same 250 genes that are rhythmic in our unaffected comparison group and then plot them in our opioid use disorder group, basically what you find is that there's a loss of transcriptional rhythms, okay? So this was our first hint that the transcriptional rhythms that we see in the nucleus accumbens that are typically rhythmic in an unaffected comparison matched cohort are arrhythmic or disrupted in our cohort of opioid use disorder subjects. These are just two examples. So each one of these dots is an actual subject. One of these genes is a circadian transcriptional repressor called CART. And then the other one is a nociceptin receptor that's highly expressed both in cortical areas, but also in subregions of the striatum. And what you can see is that in our controls, these two transcripts seem to vary nicely depending on Zeitgeber time. And we lose that time-dependent variation or that time of death-dependent variation in our subjects with OUD. Now, if we look at all of the genes that either lose or gain transcriptional rhythms in our opioid subjects, we actually found about 351 genes that actually lost transcriptional rhythms from our unaffected controls in comparing to our subjects with opioid use disorder. And we found 735 genes that actually gained rhythmicity in our subjects with opioid use disorder. And so I'm primarily going to be focusing on what we've done in this context of loss of rhythms. So what we did was we threw all those 351 genes into a blender and we asked, can we come up with any reasonable hits to predict upstream transcription factor regulators that could regulate a majority of these genes that are lost or lose rhythmicity in OUD. And our top two hits were actually known circadian transcription factors, one of which is HNF4A, which actually directly interacts with, if you remember, that BMAL1 circadian transcription factor that drives circadian dependent transcription. And then this other transcription factor that we had already been working on called NPAS2. And so this is what I call luck and serendipity. So basically what we had was we had a transcription factor that we already had some insight in our mouse models could be regulating reward and motivated behavior, but we also have a slight reduction of that transcription factor in the accumbens of subjects with OUD. So we wanted to explore this more in our mouse models. Now, Angela Osborne, who's since at OHSU now, had already published this when she was in Colleen McClung's lab in the context of cocaine rewards. So she had discovered that MPAS2 in the accumbens could be a critical modulator of conditioned cocaine reward. So we wanted to kind of take a step back and see if we could kind of walk through this more of a methodical manner. So what we did was we started with a question: do opioids alter the expression of MPAS2 in the nucleus accumbens of mice? Okay, so our prediction was that because we have a loss of transcriptional rhythms, okay, because we predicted MPAS2 as a potential upstream regulator, and we know MPAS2 typically drives transcriptional rhythms, we expected that opioids would actually reduce MPAS2 expression in our mice. So what we did was we just took B6J mice, we administered fentanyl IP in a single day or across 14 days. We harvested the nucleus accumbens and we just looked at the expression of MPAS2 along with the expression of those two other critical circadian transcription factors, clock and BMAL1. And what we saw was that chronic fentanyl, so this more repeated administration of fentanyl over 14 days, significantly reduced MPAS2 expression in the nucleus accumbens in both male and female mice. So then we zoomed out again. And so we started more with a global approach and asked, does loss of MPAS2 function alter opioid reward-related behaviors in mice? So we actually started with a mouse that has a targeted deletion of the DNA binding domain that impairs NPAS2 circadian transcription. So Steve McKnight's group, well, now 21 years ago, published in Science using this mouse, where they basically replaced this coding region for the BHLH domain with a LAC-Z. Okay, and so now we don't have a total knockout mouse, but we have a mouse where that basic loop helix domain that's required for DNA binding of NPAS2 and NPAS2 dependent circadian transcription is actually removed. So we refer to this mouse as an NPAS2 deficient mouse, not an NPAS2 knockout mouse. In conjunction with this, we wanted to develop a more translationally relevant opioid self-administration model. And we kind of started from this perspective based on the epi literature and the clinical literature, and me physically going to the addiction clinic at Western Psych and asking the attending psychiatrists what they're seeing at the ground level. So we know there's a wide availability of synthetic opioids. Combined with their high potency is really fueling the surge in opioid use and overdoses. But the demand for synthetic opioids continues to rise in states with high base rates of opioid use disorder. And mainly because synthetic opioids are cost less to produce, but they're being made in readily deliverable forms, such as wet tabs, lozenges, lollipops, and pills that can be consumed orally. So we wanted to develop an oral operant self-administration model in mouse, and there were other groups that published some oral operant self-administration models in mouse about 10 to 15 years ago. And so we kind of had to adapt what they had previously published and then change everything because what we found was it was really, really hard to get a mouse to learn two subsequent behaviors. So what we did was we trained mice to discriminate between an active and inactive lever press, where the press of the active lever dispenses a fentanyl solution into into a dipper. And then the mouse has to learn, once they've pressed that and the solution comes into the dipper, that they have to go over to the dipper and actually consume that solution. They have to associate the fact that whatever they've consumed in that oral dipper is something they like or rewarding. And so I'm not going to be showing you a lot of this pre-training data, but we tried various forms of training, including lever baiting, pre-sucrose training, and ramping up of dose. And every single one of those seems to work about the same in terms of establishing a maintenance self-administration behavior of oral fentanyl. And so we did the oral self-administration sessions for 2 hours during the light phase of the standard light dark cycle. And then we just looked at acquisition, dose response, progressive ratio responding, and cue-induced reinstatement. And so we did this in our global NPAS2 deficient mice. Okay, so I'm going to be showing you the data broken out by sex, males and females. And what we found in our male mice was that the male NPAS2 deficient mice had increased fentanyl self-administration. So over here on the left is number of rewards administered during each one of these acquisition days, so during that 2-hour session. And the red are the NPAS2 deficient male mice. And what you can see is that these NPAS2 mice self-administer more rewards over those 3 weeks of acquisition behavior. Now you'd expect, if they're self-administering more rewards, that they're actually consuming more fentanyl during those sessions, and that's what we saw. So we can measure the amount of fentanyl solution consumed at the end of the session, and then convert that to micrograms of fentanyl consumed per session over those 3 weeks. And NPAS2 male mice tend to consume more fentanyl over those 3 weeks. And they also had a shorter time in terms of days to criteria. So stable responding over 3 days of 2 active lever presses to 1 active lever press. So the male NPAS2 mice seem to have increased opioid seeking behavior. So this was actually in contrast to what we saw in our female mice. So basically what we saw in our female mice was that females tended to self-administer more fentanyl, but the effect was regardless of genotype, okay? And so if you actually plot where the male's max is for the wild type, female wild type animals tend to self-administer more fentanyl. But MPAS2 deficient females tend to self-administer at the same level of MPAS2 deficient males or a little bit lower. And we also see that in the amount of fentanyl actually consumed. So we can also look at craving and relapse-like behavior in our models. And so this is just, again, males and females broken out. So we did progressive ratio and dose response. And then we also looked at the break point across various doses of fentanyl using this oral self-administration. And if you're used to looking at intravenous self-administration data, this is not as clean as intravenous self-administration data, but we did see an overall genotype effect for increased breakpoint or increased craving motivated behavior to self-administer fentanyl. We can somewhat extinguish this behavior in our wild type and NPAS2 animals over the course of 10 days. And then on day 14, expose the animals to a various set of cues that were previously associated with dispensing of that fentanyl to see how they respond. And so both wild type and NPAS2 animals respond to those cues, expecting that they're actually going to receive a reward. Okay, so they have increased cue-induced reinstatement. And in our females, we did not see any difference between our wild type and NPAS2 animals, but we did see an increase in cue-induced reinstatement behavior. So we also looked at development of tolerance, opioid tolerance to analgesia and fentanyl withdrawal. So we did 2 dose response curves where we exposed animals to varying doses of fentanyl and then tested tolerance in between on days 4 through 9 and then withdrawal on day 13 and 14. And we didn't really see, and then measured the response to a hot plate using tail flick latency in both males and females, wild type and NPAS2 deficient animals. And we really didn't see any kind of dramatic effects on fentanyl tolerance to analgesia. What we did see is that there was about a modest rightward shift in male MPAS2 mice in fentanyl tolerance. So if you compare this light blue to dark blue lines, okay? However, when we looked at withdrawal, so this is naloxone-induced fentanyl withdrawal on day 13 and 14, what we saw was kind of this very large naloxone-induced withdrawal effect that seemed to be sex-specific. So we saw an increase in withdrawal score that's comprised of a number of different behaviors, including teeth chattering, diarrhea, jumping, seizures, and so forth, in relation to their male MPAS2 counterparts, but also in relation to wild type animals. So far, I've shown you that MPAS2 deficiency leads to increased fentanyl seeking, craving, and relapse behaviors in male mice. There may be some sex-specific effects on that self-administration behavior, but there are definitely sex-specific effects on the fentanyl tolerance and withdrawal behaviors. And the withdrawal effect seems to be particularly pronounced in female mice. So now that we started more global and wanted to ensure that the actual MPAS2 deficiency modulated or regulated these opioid-related behaviors, we wanted to kind of come back to the nucleus accumbens where we had this predicted transcription factor in our human data and where we already kind of knew where MPAS2 was mostly enriched. So we know a lot about the nucleus accumbens. This is not meant to be dogma in any sense, But the nucleus accumbens is primarily made up of GABAergic medium spiny neurons that express either D1 or D2 receptors. And these neurons send projections throughout the brain. But D1 medium spiny neuron activation typically promotes drug-related behaviors, and D2 MSN activation antagonizes drug-related behaviors. And there's a lot of nuances and contradictory evidence in this context. But for the purpose of this, we wanted to frame the question, is NPAS2? So if you had a loss of NPAS2 function that drives opioid reward-related behaviors, is NPAS2 actually enriched in D1 cells in the nucleus accumbens? Well, like I alluded to, we already had some insights for this. So back in the year 2000, in that NPAS2 deficient mouse paper in Science from Steve McKnight's group, they looked at LAC-Z expression throughout the whole mouse brain. Okay, so this LAC-Z the LAC-Z cassette is actually driven by NPAS2 expression, and they found this black hole in the nucleus accumbens. And Angela Osborne, the paper that I mentioned previously, had shown that NPAS2 might actually be enriched in D1 neurons. So we wanted to kind of recapitulate these findings using a different, more sensitive approach. So we turned to RNA-scope-based labeling, where we have these fluorescent probes that can tag NPAS2 and various other genes. And we imaged and labeled the entire striatum and then did cross-sectional counting. And basically what we saw was that NPAS2 was indeed more expressed, preferentially expressed in D1 positive neurons, about 60% of the total neurons, okay? So we wanted to go back to our human data, okay? And to see if this if our human data could steer us again to D1 neurons. So we did single cell sequencing in macaques, and this is in collaboration with Andreas Fetting and Bill Stouffer at Pitt. We did single cell clustering in the accumbens and other brain areas. And then we took that single cell data, those transcriptional profiles, and aligned it with our gene signatures in the nucleus accumbens in our subjects with opioid use disorder. And then did a very, very simple hypergeometric t-test where we were testing, are these genes enriched in our subjects with opioid use disorder versus our unaffected comparison group, okay? So this is kind of bringing it back to the human to see if we have some converging evidence. And what we see is that we actually have a high level of enrichment and things changed in D1 medium spiny neurons in our subjects with opioid use disorder. So we went back to our mouse and asked, does NPAS2 regulate fentanyl reward via action in D1 cells and nucleus accumbens? And so the way we did this at the time when we were designing these experiments, this seemed very complicated, and it is very complicated, and there are definitely other ways to do this now. But we used CRISPR to design CRISPR knock-in mice, where we were using an intersectional genetic strategy called split Cre approach, where we can actually drive different Cre constructs with different promoters, okay? So what we did was we created multiple lines of mice where the Cre-N terminus is driven by a DRD1A promoter or a DRD2 promoter. And then we created another line of mouse where the Cre-C terminus, which is attached to an ERT2 sequence that's inducible with tamoxifen, is driven by GPR88. Now, GPR88 in the mouse is highly enriched in the striatum, okay? So what you were supposed to get when you cross these lines, line 1 with, or line 2 with GPR88, is the reconstitution of Cre only in D1 or D2 expressing cells in striatal neurons, okay? We also designed another line, the NPAS2 flox mouse, where we flanked the part of exon 1 that codes for that basic helix loop helix domain like our global MPAS2 deficient animal, that when crossed to our full Cree mouse and then induced by tamoxifen, is you get D1 specific or D2 specific striatal MPAS2 deficiency. Okay, and there's other various labels in here that I don't need to go into, but GFP and SuperBlue can label these cells where we've had actually successful Cree recombination and so forth. So this is just kind of one example using base scope labeling, where you can actually design little probes that target a specific sequence that's attached to a fluorophore. And what you'd expect to see is Cree expression only in the striatum and only in D1 cells. And this is just kind of a zoomed out perspective, but basically what we see is Cree expression only in the striatum. And you'll have to trust me, that's also only in D1 cells. So this is just some preliminary data. So we did D1 targeted MPAS2 deficiency in male mice. We've also done this in females, but that data is not ready for prime time yet. And so on the left here is our global MPAS2 deficient mice, males and females. And we did kind of a quick and dirty condition reward assay called condition place preference, where you pair drug or saline on opposite sides of the 3-chamber box. And then over a number of pairings, ask the animal, where do you like to spend most time? Most animals who've gotten a rewarding substance tend to spend more time on the side where they received drug. And so in our global MPAS2 deficient animals, males and females, the genotype actually increased the condition reward response, so the amount of time they spent in the fentanyl pair side. And then when we looked at our D1 and D2 deficient animals, we actually recapitulated that increase in condition fentanyl reward only in our D1 MPAS2 deficient animals and not in our D2 MPAS2 deficient animals. And so right now we're working through the self-administration behavior and really fleshing this out, but it seems to be consistent with what our kind of converging data from both human and our global MPAS2 deficient animal is pointing us. So using time of death analysis, we were able to recreate transcriptional rhythms in the human brain. And then we were able to actually use that data to look at things that might be upstream regulators or mechanisms that could guide some of these opioid reward-related behaviors. We identified MPAS2, and we've explored in both global MPAS2 deficient and D1 specific ablation of MPAS2 that both of these things seem to increase opioid reward behaviors in mice. So what are we doing now? So we have a lot of different things going on in this arena. And so I'm just going to really briefly touch on some of the things that we're doing in this arena. So we believe that MPAS2 could be a key molecular mediator in D1 neurons that could explain the detrimental effects of opioids or fentanyl on sleep. We also have some data to suggest that when you rescue MPAS2 in a cell type specific way, that you actually augment the response of animals that have been self-administering fentanyl for weeks, if not months, in their response to buprenorphine. So we've started to characterize sleep in our MPAS2 deficient animals. Now, Paul Franklin's group and a few other groups have actually looked at sleep architecture in a similar mouse, but we use sleep polysomnography in mice, and we just looked at non-REM and REM sleep and classified REM and non-REM sleep in light or dark phases of the light-dark cycle. And what we found is that MPAS2 deficient animals have about a 30% reduction in REM sleep. We can also disrupt sleep in a very sleep state specific way. And so this is a collaboration with Chris O'Donnell where we've designed an air puff system where we can record sleep in real time and then interrupt when the animal goes into non-REM or REM using basically air puffs to the animal's face. And this is just kind of proof of principle data showing that we can almost totally ablate REM sleep using this model. We have some preliminary data to look at this interaction of sleep and opioid administration. And I don't need to take you through all of this, but basically we did a within mouse design where we have a baseline sleep recording and then various sleep recordings pre and immediately post the administration of fentanyl. And we just compared our acute administration of fentanyl, so a single injection, versus an injection that occurred over 7 days and then measured sleep. Now, we did this both in our wild type and MPAS2 deficient mice. We have a lot of data, but probably the coolest part of this data is that when you look at the chronic administration group, and then you look at the first 24 hours after the last fentanyl injection, and then measure sleep across those 4 days of withdrawal, okay? What you actually see is this progressive loss of non-REM sleep that occurs over days of withdrawal, okay? So these animals on withdrawal day 4, that's labeled WD4, haven't seen fentanyl in 4+ days, okay? And you still get this 15 to 20% reduction in non-REM. Now, why do we think this is important? Because we know that during abstinence, people who chronically administer opioids display reduced total sleep duration, increased fragmented sleep, reduced REM sleep, like we've demonstrated in our MPAS2 deficient animals, and increased wakefulness. And so when you have a reduction in non-REM, actually what's happening is when those animals transition or are trying to transition to non-REM, they're waking up at the end of a REM episode. So this is actually reflecting possibly a hyperarousal or increased wakefulness state. And so again, we think MPAS is this molecular switch. We're looking at how MPAS actually governs neuronal activity in the nucleus accumbens and other brain regions, but what MPAS2 is actually directly transcriptionally regulating, okay? And we're doing a lot of different approaches to further characterize MPAS2 actions and a few other transcription factors we found in our MPAS animals. So in the last couple of minutes, I think I have a couple of minutes. Cameron, do I have a couple of minutes? Okay.
Yes, sorry.
Okay. All right, sorry. So I wanted to present this because we're really trying to advertise this because we want people to collaborate and use this. So one of the things that's a problem in neurobiology and neuroscience is really kind of bridging the gap from genetics to circuits to behavior. And one of the limitations is, of course, tools and methodologies that allow us to do that in a comprehensive, complete way. So myself, with two collaborators, Zach Freyberg and Alan Watson, who's at the Center of Biologic Imaging at University of Pittsburgh, have designed and utilized this new approach called ribbon scanning confocal microscopy, okay? So ribbon scanning confocal microscopy is much different than kind of a conventional imaging approach that relies on a stop and shoot method, okay? So every time you take an image, you have to move another XYZ plane, take another image, move, take another image, and then you have to stitch all of these images back together to then recreate or render the complete or intact tissue that you're imaging. What ribbon scanning does is actually we can scan and image simultaneously in any direction that we want. And so now we've limited the amount of times we actually have to stitch those images back together. Why does this matter? Well, it increases our resolution and also reduces our time. So we've actually combined this with a modified cubic based tissue clearing approach to clear whole brains in mouse. And we took a proof of principle approach where we crossed a dopamine transporter Cree mouse with a TD tomato flox mouse. And so anywhere you see TD tomato or GFP is a dopamine neuron. And we imaged the entire intact whole brain of a mouse. And we've started to map individual dopamine neurons and their projections throughout the whole brain, okay? Now what you can see is that we can go from whole brain to single cell in the same intact whole tissue. Now, it took us about 18 hours to image and render this video. So we can actually do this in less than a day with a single color, okay? What we're working on now is computational pipelines to image these large data sets. So each brain imaging data set is about 2 terabytes, okay? So we're leveraging the infrastructure from the Pittsburgh Supercomputing Center and the Center of Biologic Imaging to analyze these data sets. And our hope is to go from kind of simple rudimentary analysis of counting cells, okay, with a particular reporter to be able to trace the projections from those cells throughout the whole brain, and then map those circuits back to the Allen Brain Atlas at 200 nanometer resolution, okay? So right now, that is what we're currently working on. Now, one of the ways that another way that we've leveraged this approach is actually a collaboration with Carl Dijsselrath's lab using this intersectional genetic approach called Intersect 2.0, which is based on Boolean logic gates, okay? So you have a flip Cree system where the flip and Cree are driven by 2 different promoters. In this case, we have a mouse that is TH flip, okay? So wherever tyrosine hydroxylase is expressed, we have flip expressed, and then VGLUT2, so the glutamate transporter that drives Cree. When you cross these animals, what you get is animals that express flip Cree in TH and VGLUT2 positive cells. But what you also get in that same animal is you get M-cherry labeling of TH only neurons, okay? And then EYFP labeling of those TH and VGLUT neurons. So in the same animal, we can look at TH only neurons relative to neurons that express both TH and VGLUT2. And we applied this with our imaging approach. We imaged the whole brain. So wherever you see red are actually projections from dopamine neurons. And then we quantified the percent total cells in the ventral tegmental area of the mouse that express only TH versus the amount of cells that express TH and VGLUT2. And this is kind of very rudimentary, but it's a proof of principle. And as you'd expect, there's a higher number of cells in the ventral tegmental area that express TH only versus the cells that express TH and VGLUT2. And we're doing a lot of this to characterize what the differences are of those TH and THV-VGLUT2 dopamine subpopulations in the mouse using this approach and viral approaches. The other thing we can also do is take these cleared brains and soak them in antibody baths, okay? So you don't necessarily need your favorite reporter mouse. You don't need a sophisticated transgenic mouse. You don't need to create a mouse. What you need is a really good immunolabeled antibody. And so, again, this is just proof of principle where we soaked mouse brains in solutions that contained antibodies to label GFAP, so astrocytes, and then imaged the whole brain. And so what we're working on now is actually looking at whether our immunolabeling across every axis of the brain and our imaging is uniform, okay? To make sure that we don't get differences in immunolabeling penetration of the antibody, and so forth. But what you can see is that we go from whole brain to beautiful astrocytes. And in doing some of these pilot experiments, we've actually discovered some really, really interesting new biology that I don't have time to get into. But you can see we can do this in whole brain to the single level. Now, one of the things we wanted to do was actually take our ribbon scanning confocal microscopy approach and compare it directly to what was available for light sheet microscopy. So I'm sure a lot of people have heard about light sheet microscopy. You can image the same tissue from different angles at a similar 200 to 300 nanometer resolution. And so the Center for Biologic Imaging bought a light sheet for us, and then we combined side by side our ribbon scanning confocal microscope approach to light sheet. And I just want to point out one thing, is that the light sheet microscopy approach cannot reach or exceed our current resolution and depth of the ribbon scanning confocal microscope. So what we actually had to do was undersample using our ribbon scanning confocal microscope to compare apples to apples. Okay, so in each one of these cells is actually days to complete. So we imaged brains with one color all the way up to 4 colors. And what you can see is that we get faster with the more colors and at a higher resolution than light sheet. So we can image an entire brain up to 3 colors in about 4 days, whereas light sheet takes about 14 days. So it's scalable, diffraction limited resolution. We can label multiple colors. We can actually image and render 3D whole maps in less than 24 hours. And we can right now do about 24 brains per week, although that is going to increase. And at this current moment, we're relying on the computational and storage infrastructure from the Center for Biologic Imaging in the Pittsburgh Supercomputing Center, and the infrastructure that Alan Watson at University of Pittsburgh has set up. So with that, I want to thank my lab members that contributed to this work, particularly Kelly Barco and Samoon Kim, Roy Fan, and Stephanie and Micah. I also want to thank a few collaborators that directly contributed to this work, including Zach Freiberg and Joel Glosher, David Lewis, who is my former chair, Chris and Andreas, Marianne, Sine, and I have a multi-PI R01 where we're forming a new cohort of opioid use disorder subjects. We're actually going to look at different omic scales, all the way from single nuclei up to synaptosomal fraction, proteomics, and bulk proteomics in about 200 subjects across 4 brain regions. And then George Chang, who helped with some of the computational analyses, and of course, Alan, and then current funding for my lab. So with that, I can take some questions.
Wonderful talk. Thank you, Ryan. Okay, we do have some questions. Haley Thorpe asked, is there any compensation by clock in your MPAS-2 deficient mice? I'm also wondering how your results with MPAS-2 deficient mice compare with the clock deficient mice. Are they broadly similar in the reward system changes, or are their hallmarks specific to MPAS-2?
Yeah, Haley, great question. So the simplest answer is we have looked at clock compensation in the nucleus accumbens in global MPAS-2 deficient animals, and we do not see compensation of clock, okay? We've done that by looking at protein competition assays, chromatin immunoprecipitation, co-IP, and so forth. We just don't see a compensation. Now, that's very, very different than when you knock out clock in, let's say, the ventral tegmental area. In that case, when you knock out clock, you actually do see some compensation of MPAS-2. And DeBrine's group, Michael Menaker's group, a number of years ago, had shown that there is reciprocity of clock and MPAS-2 when one of them is knocked down and so forth, but it seems to be brain region specific. And then the second part of the question, are they similar? No, so we don't see similar opioid reward changes in the clock delta 19 mice that Colleen McClungham has done a lot of work with. But again, we haven't done an extensive set of study. We've done morphine locomotor sensitization, we've done condition place preference and so forth, but we don't see the same increase.
Okay, Julie Blendy asks, how are opioids regulating impasse? What upstream signaling may be involved?
Yeah, so we don't know yet. We don't know. There is one paper showing that when you kind of tune D1 through pharmacological application in primary striatal cultures, that you get an increase or decrease of MPAS2 expression in those cells. We have that is definitely something that we're interested in, but we've been mostly focused on everything downstream of MPAS rather than everything that's upstream.
Next question is from Razi. Very interesting talk, Ryan. So how does your sex difference in mouse study relate to human postmortem study? Do you have sex differences in humans? Also, in light of the mouse study, can you conclude that disruption in circadian rhythmicity predisposes people for OUD, or circadian arrhythmicity consequence of opiate consumption? Thanks. The age-old question.
So I'm going to answer this backwards. So in our human postmortem studies, we obviously cannot directly parse out like our mouse models, the consequence of chronic opioid use versus the predisposition or vulnerability. Okay. But we are forming human postmortem cohorts of subjects with opioid use disorder where we're trying to ask various parts of that question, right? So we're forming cohorts where you can actually select subjects who have a diagnosis of opioid use disorder who are in quote unquote remission. We have extensive medical records on every subject that we collect, and we have extensive 12-month family interviews, okay? So that we can put subjects into, they've had a diagnosis with OUD, they're not actively using, or at least no active use. They have a clear blood tox when they die, and they've died of something else, versus subjects who have opioid use disorder who may have died from an overdose or has a positive blood tox screen of opioids and other substances. So we don't know yet. That's kind of part of the reason why we're trying to bring it back to our animal models, but we can chip away at parts of that question. Sex differences. In our cohort of 20 subjects versus 20 subjects, we don't have a lot of overt sex differences, okay? However, when we first designed the cohort, we matched based on sex, but we did not design in other opioid use disorder clinical characteristics, but we did not design the cohort to ask a sex difference or sex specific question. So that is definitely something that we're going to be doing in future human postmortem studies.
Okay, one more question regarding sleep studies. Wouldn't you predict that during withdrawal from opioids, animals will have disrupted sleep, especially NREM, during which delta sleep occurs? That would be based on the assumption that opioids increase NREM and help animals go into deep delta sleep. Is it known that this is the case?
Yeah, so there are a couple of other groups looking at the impact of opioids on sleep architecture in rodent models. And we're all finding kind of different things depending on how we expose and how much we expose and which opioid we expose the animal to, okay? Our original hypothesis was that we were actually going to see a reduction in REM sleep because the animals were actually going to be incapable of transitioning from non-REM to REM, okay? So yes, I would agree with you. That was kind of our original hypothesis. That's not what we found in our first set of pilot studies. We found this really dramatic reduction in non-REM. I didn't show you the REM data, but we don't really see any reduction on on REM, so to speak. What we do see is reduction in non-REM, increased in awakenings, shorter sleep bouts, and so forth. And so that's kind of consistent with this idea that, at least in our chronically exposed animals, that chronic opioids does lead to this kind of persisting sleep change. What was really interesting in our data that I just want to come back to and highlight is that all of the things that we saw in non-REM were in about a 5 to 10% range on the last day those animals were exposed to opioids, okay? But when we measured sleep during that first 4 days of withdrawal, it got worse, okay? So what we're really interested in now is that progressive loss of sleep quality, or that progressive change in sleep architecture across withdrawal that isn't really noticeable when the animal's exposed to or has been exposed to in opioids in the last 24 hours. Does that make sense? It seems to be something that's changing during withdrawal when opioids are removed.
Okay, great. Any other questions? Going once, going twice. All right, let's thank Dr. Logan again for this talk and congratulate him on winning this prestigious award. Thank you, Ryan.
Thank you, Cameron. Thank you, everybody.
And my only announcement is we will have a new webinar link for the 1 p.m. keynote address by Dr. Hopi Hoekstra. So please make sure to use the new link and we'll be reconvening at 1 p.m. So I'll see you all then. Thank you.